today someone visited me and said that they didn't think much was on this exam. So I was like, well, that's a personal gift from me to you. An exam with nothing to study. So I hope you all feel that way, because then you can all rock it. Okay, let's see. Now, a little bit. No one's here today. I know, on the exam, everyone's like, get out of my seat. Okay. Hopefully people have realized the exam is next Monday. Sorry for the confusion about Thursday recitation. I was just trying to give people the option to go to whatever recitation they wanted. Thursday recitation is not canceled. It is still happening. Please attend it if you cannot attend any others. I just want, sometimes with these review practice problems, it's, I don't know, you can ask people at recitation today. It might be nice to have me there, but Dr. Texel does a fine job too. If there's any confusion afterwards, just come see me in office hours. Or not, actually, because there's no more office hours. So then email me and with any clarification. Okay, so this is what we have done so far. We've talked about signaling. I went through a whole bunch of different pathways and probably overwhelmed you, but just that was kind of the introduction, and then we will go through them towards the end again, and I'll point out the different, how they're examples of different things that we've learned. We kind of started at the top with the signaling molecules, and then talked about the different types of receptors. And then we talked a little bit more specifically about signaling through GPCRs and RTKs, and we're going to be talking about kinase-associated receptors. And then we've been talking about some of the more important intracellular signaling molecules, such as RAS, and MAP kinases, and scaffold proteins. And so I want you to just start to try to organize these things in this kind of, categorizing, like here we're at the top, these kinds of proteins. I have nuclear receptors. These kinds of proteins bind to these kinds of receptors, and they do these things. And, you're more likely to have these small intracellular mediators with GPCRs versus the RTKs or kinase-associated receptors. You can start to kind of make your own little categories for each thing and put all these pieces of information somewhere. And I'm hoping to finish today and have Wednesday for review. Last year I was able to kind of organize it all for everyone, and they really liked it. So I'm going to see if I can do that on Wednesday, and then we can ask questions. Okay, so we were finishing the other day with RTKs, and we were talking about how receptor tyrosine kinases activate RAS, and that helps to spread the signal to multiple pathways, and they do so through recruitment of an adapter protein to the phosphorylated receptor, which then recruits a GEP. So it's regulated at the point of whether or not the GEP is recruited to activate RAS or not. And so there's an example in the book of how all of this was discovered. So I thought I'd put something up about that because I liked this picture of the compound I. Isn't that cool? So it's made up of 800 identical units, and it develops from cells in an epithelial sheet that are recruited in a specific order. And so there's this one cell that recruits these cells, and it's the communication. So this ends up being your ommatidium in the end, and ommatidia. And it's the communication between the different cells in each individual ommatidia that that tells the cell what genes to express to become a photoreceptor, or to become that cell type. So each component in the pathway was developed through screening of different Drosophila mutants, and they have all these very cool names, like Son of Sevenless and Darkness, and all the, I don't know, sci-fi lovers work in Drosophila. And so, but basically what we're showing here is the communication between a receptor and a ligand, but where it's not a secreted molecule. Again, I keep telling you that it's not always some kind of secreted molecule that's binding to a receptor that's being brought into the cell. That's very much not what's happening here, which is why I put it up. So here you have your R8 cell, and the last cell that's recruited is the R7 cell. And it needs to tell R7 cell to be an R7 cell and to participate in becoming the ommatidium. So there's this receptor tyrosine kinase, and it binds to this other receptor on the R8 cell, and it's through that receptor ligand interaction that the signaling occurs that tells the R7 cell what kind of cell to become. Okay? And so the way so this is an example of using, you have a receptor that has the intrinsic kinase activity, and then you get phosphorylation of the receptor, which creates a docking site, and then you get this adapter protein recruited that brings in your RAS-GEF, and you have RAS hanging out there, and RAS gets activated because of the recruitment of the GEF, and that kind of turns on the signaling that helps to determine the fate of the cell. Okay? And so I thought that was kind of a nice example in the textbook. Any questions? Yes?
I guess there's a lot of RAS in the membrane, so things they have to be in control.
So the comment was there must be a lot of RAS in the membrane because it must be in close proximity to receptors for the gap to be close to RAS, right? Is that what you're saying? Yes, there's probably a ton of RAS, but don't forget that membrane is not this static environment. It's fluid, and things are always moving around also. Yep. Any other comments? So this is just showing a little bit more closely that the interaction between these two different cells activates the gene expression required for development of the R7 cell into the R7 photoreceptor. Okay, so this is an experiment that people do to look at RAS activation, and it's called FRET, which is fluorescence resonance energy transfer. It could sound complicated, but really it's just about looking at excitation of fluorescence when two proteins are close together. So you'll have RAS. So here's RAS. It's normally GDP bound. And so what they did is they engineered it to be tagged with YFP, yellow fluorescent protein. And you can excite it, and it emits a yellow color. And then they have red fluorescent GTP in the cell. And so when GTP is bound to RAS, the energy is transferred and it changes color. And so that's how you can look at RAS that's active in the cell. And so people use this because you might want to study how long is RAS active after you add your signaling molecule to your media. So these could be cells, and where you're studying, in this example, EGF, epidermal growth factor receptor, and you'll add EGF, and you'll look, and you can actually use a microscope and look at the color of the spots on your cells. If they're yellow, then they are GDP bound because that's just the color of RAS when it's not GTP bound, so they're inactive. And when GTP binds, so once RAS is activated, you're going to get a color change, and then they'll become red. Okay? Very cool, huh? And so what they did, what they're showing in this experiment is just that RAS activation is really quick, and it's really short and transient. And you can see that because your number of activated RAS, after you add your EGFs, it goes up really fast and it comes down within minutes. And so that's important, right? Would you want your RAS to be on for a really long time? Probably not. And so the activity of RAS is quickly reversed by the activity of phosphatases. So who knows what phosphatases are? Oh, so many people want to tell me. I love it. Everyone shout it out. Remove phosphate groups. Good. You did your homework. Very good. Great. So just as important as kinases are to add phosphate groups to things, to change the shape of the protein, to activate something, recruit something, whatever, phosphatases are equally important in removing those phosphate groups to either inactivate or activate or whatever's going on. Okay, very cool. Does anyone have questions about this? I've always wanted an excuse to do FRET. It's not limited. This method is not limited to GTP binding of RAS. You can use it to look at any protein-protein interaction because proteins need to be, or biomolecules need to be very close in order to transfer the energy to change the color. So I think that is super cool. Yay? Okay, I see some people nodding. Okay, and that was that. Okay, so let's talk about PI3 kinase. So PI3 kinase, it can be activated by a whole bunch of things. It can be activated by RTKs. It can be activated by GPCRs. It can be activated by RAS. But it is a kinase, so it's going to phosphorylate something, and it phosphorylates inositol phospholipids in the membrane, and that creates docking sites for more proteins. So notice we keep phosphorylating things and creating docking sites for things, right? Is that a theme here? It's all about recruiting the proteins that you need for whatever you're doing. And it's dephosphorylates, so PI3 kinase phosphorylates inositol phospholipids, and then those are dephosphorylated by PTEN. Well, PTEN is the most common one, you should know that it's highly mutated in cancer. So remember, when you get phosphorylation of these inositol phospholipids, then you're creating docking sites to recruit signaling proteins that participate in signaling, and so you need to dephosphorylate after to turn that off. And so if P10 is mutated and it can't dephosphorylate, what might be the outcome? Cancer. You guys are trained so well. Cancer. No, well, so if you can't dephosphorylate, you're going to never you lose your regulation. So through this whole unit, I want you guys to constantly be thinking, what's being regulated here? How is it being regulated? And it usually has to do with something being phosphorylated, something being dephosphorylated, some protein being recruited to switch out a GDP for a GTP. There are some themes that are happening again and again, right? Right? So PI3 kinase is involved in cell growth and survival, and it's not surprising that this pathway, or that this kinase is highly involved in different processes in cancer. Cancer! So this is one pathway. This is one pathway that's involved with PI3 kinase. That's involved with cell growth and survival, where you'll have some kind of survival signal. I think we said in the book insulin is a big one. Will bind to a receptor, and then you'll get recruitment and activation of PI3 kinase. And it will phosphorylate these phospholipids, which will create docking sites for these other proteins with these pH domains. What do you think pH domains bind to? Who wants to guess what a pH domain binds to? Yeah. A phosphate, but in the context of these inositol phospholipids. Yeah, so like PIP2, yeah. So these are proteins. I keep thinking that people are still confused about domains. These proteins, PDK1, AKT, they have pH domains, meaning part of the protein likes to bind to phosphorylated inositol phospholipids. Okay? And so they get recruited up here, and then they get activated, and then they get phosphorylated, and then AKT will dissociate, and it will phosphorylate BAD, which inhibits BAD. Of course, you want to inhibit BAD. And then you get this apoptosis, inhibitory protein activated, and it can inhibit apoptosis. Okay? That's probably not a good thing in cancer, right? You don't want to inhibit apoptosis in cancer. You want to promote apoptosis. So it makes sense that rapamycin, which is an mTOR inhibitor, is a cancer treatment, which is actually not going to inhibit apoptosis. It's going to promote apoptosis. So it inhibits the inhibition of apoptosis. So it promotes apoptosis, which is what you would want to do if you were designing a cancer drug, right? Would you want to promote apoptosis or inhibit it? Promote it. Good job. Yeah. Okay. So just when you thought you understood it all, I'm going to tell you that there's a lot of crosstalk between these different pathways. So I'm showing you a GPCR here, and I'm showing a receptor tyrosine kinase here. And you can have different signaling molecules binding to different receptors that might still end up activating similar pathways that result in activation of similar kinases that will target similar or different proteins. And so you should just kind of appreciate the fact that it's not just this linear process, and that often one pathway will activate another pathway, and it's all very specific to the cell at the time. Okay? How do you feel about that? Does that make you nervous? It makes you nervous for the exam. So here, right, because here you have a G protein that's going to activate adenyl cyclase, which activates cyclic AMP and PKA, and that activates gene regulatory proteins. But at the same time, you can also have a G protein that's going to activate phospholipase 3, which can go either in this direction or this direction, or there's a lot going on. That's why whenever you look at these signal transduction pathways, there's a million proteins with arrows in a million directions. And that's why it's fun. It's good for making exam questions. Okay, so we've been talking about receptor tyrosine kinases. These are these kinases that have intrinsic kinase activity, right? Yeah? No? Yeah? Okay, so now we're going to talk about tyrosine kinase associated receptors. This keeps confusing people. I don't know why. It probably is because of the words. Just remember, one has intrinsic kinase activity, one binds to and uses the kinase activity from another protein kinase. Okay? So here's an example. Remember, this is right out of the JAK-STAT pathway, where you have the receptor and it's associated with a kinase. Most of these kinases are members of the Src family, And they all contain SH2 domains and SH3 domains and bind to cytoplasmic domains of receptors. So you might be saying, wait a minute, I know what an SH2 domain, but what I know what that is, but what's an SH3 domain? SH2, remember, binds to phosphotyrosine. SH3 binds to proline-rich sequences and other proteins. So basically, it's a protein-protein interaction domain. Why do you care? It's just something that makes it part of the same family. There are all these Sark family of kinases. If a protein is going to be part of a family, it's going to have things in common with other proteins within that family. Okay, so this is where we kind of started, right, a few weeks ago with the JAK-STAT pathway. And so hopefully we're going to go over it again. Hopefully it makes a bit more sense than it did the first time around. So this was an example of signaling through a tyrosine kinase associated receptors, right? So you've got your receptors that are always bound to these kinases, and then you'll have a signaling molecule that will bind and bring the receptors together, and you'll get the cross-phosphorylation, which will activate the kinases, and then you'll get phosphorylation of the cytoplasmic tail of the receptor, which will create docking sites for other proteins, in this case STAT1 and STAT2, which will then get phosphorylated, and then they'll detach from the receptor and dimerize, and that creates some kind of NLS type of motif, which allows it to enter the nucleus, where it will bind to the DNA and activate gene expression. Okay. So this is involved in the antiviral response. So obviously you would need to turn this off at some point, right? Or do you want to keep this on even when the virus is gone? You guys are like dead people today. I can't tell you the question. Top secret questions here. Okay, so this is part of the antiviral response, right? Because when you have a virus in the area, cells around will be secreting interferons, and that will turn on gene expression, and all these genes that are associated with fighting a virus will be expressed. But you don't want to keep that on forever, right? Or do you? No, when the virus is gone, you need to turn your immune response off. And so I'm just wondering if you guys remember how you might turn this off. Right? Because we've got all this going on here. Okay. How do we turn it off? Okay, I heard something about dephosphorylation. Okay. Something about removing, so you can dephosphorylate here. I guess if there's a phosphatase, that might prevent interaction here. What else might turn this off? Huh? IKB, that's a different pathway. Receptor endocytosis, that could also, right? So if you endocytose and you downregulate your receptors, yep. What else? Yep, you can get ubiquitination and degradation of transcription factors, which we have not talked about yet, but that's a very common way of shutting off signaling, is to ubiquitinate and degrade different signaling molecules involved in the pathway. Yep. Anyone else? Yes. GTP, is there GTP in this pathway? No, but in the case of GPCR, GTP hydrolysis will shut off. So in this pathway, you gotta be careful and keep straight about what kind of pathway you're talking about. So I heard IKB, that's specific to a certain pathway. GTP hydrolysis, that's for a different kind of pathway. So start to get organized into what components are in what pathway. Right, so tyrosine phosphatases modify or turn signals off. So that was the first answer I got, and that was right. So you can have cytoplasmic phosphatases that are just these soluble proteins in the cytoplasm that will interact with and remove phosphates in a regulated way, right? This has to be regulated also. Or sometimes they are plasma membrane bound, and that it seems to be unclear as to why they look like receptors. And so this is showing the tyrosine phosphatase domains in the red. So how might a phosphatase modify a signal? So we talked about turning off a signal. So how would it turn off and how would it modify a signal? And what do I mean by those two things? What does modify signal mean? Change it. And how might you change it? Okay, so if you remove a phosphate group, you might change the binding site. So you might change what proteins are being recruited, right? Right? And how would it turn off the signal? The same thing. It just depends on what you're removing from the system. Okay? So remember, all these different proteins are recruiting to make the signaling complex. Any thoughts? Any questions about phosphatases? Phosphatases are going to come up again. They will. When we do cell cycle. So it's definitely important to make sure you understand them this time around. Actually, a lot of this stuff keeps coming up. You notice we keep talking about endocytosis. We keep talking about nuclear localization. Yes?
So when you see how we turn off the signal, that means
How would it turn off the signal? So let's see if anyone else wants to answer. How would a phosphatase turn off a signal? Well, let's get more specific. What does a phosphatase do? Removes a phosphate group. So makes a conformational change. I guess vaguely, yes, but more specifically, it removes a phosphate group, yes.
Like in a that one, like if you remove a phosphate, anywhere at the top in the early one, would it prevent the following?
Right, so in the JAK-STAT pathway, if you remove a phosphate anywhere up here, it will prevent the pathway from moving forward. Yeah, because so for here, if you remove this phosphorylation, then those kinases won't be active and they won't phosphorylate here. If you remove those phosphates, then you won't be able to recruit stats to be phosphorylated and they won't be able to dimerize. They won't be able to go into the nucleus and then they couldn't activate gene expression. Does that make sense? Yeah? Yeah. Any other questions about this? Yes?
So does it just depend on what phosphate it's removing as to whether it's modifying it or turning it off?
Yeah. So the question is, does it depend on which phosphate is being removed as to what the outcome is, whether it's modifying or turning it off? Yes, and that's why I think it frustrates students sometimes with the phosphorylation and phosphatases and all these modifications, because it's not always a definitive outcome, like you phosphorylate, you activate. It's not like that. It depends on what's being phosphorylated where, and what's being recruited, and what's like the specifics of what's happening. Which is, I don't know if you guys remembered, I think earlier in the semester, I put up a picture of P53 and I showed you it's phosphorylated in a gazillion different places, and they all have a million different outcomes. And so just, you have to like open your mind to the possibility that it's doing many different things. Yeah?
Is that probably like modified signal thing? They just have a phosphorylated in different places?
Yeah, so that's kind of the modified signal thing, exactly. So depending on where you're phosphorylated, you're going to recruit different proteins, which might activate different parts of a pathway or different pathways. Yep. You might phosphorylate in one place and recruit the adapter for activating RAS, right? And in another case, it might recruit something else for doing something completely different. Okay, good. Anything else? Okay. So this is SMAD. These are SMADs. I always like them because it's a weird word. SMAD dependent TGF beta signaling. So this is another pathway, new and exciting. And this is another example of signaling through a receptor kinase. So it has its own kinase domain, but it has a serine threonine kinase domain, but still it has intrinsic kinase activity. It just so happens that it phosphorylates serines and threonines rather than tyrosines, whatever. And so this pathway is involved in development, tissue repair, and immune regulation. And it's our first example, well, not really, but it's my introduction to latent gene regulatory proteins for you today. So normally you'll have a ligand bind to a receptor that will bring receptors in close proximity, right? And so there's two. This is not a homodimer; it's a heterodimer, meaning that there are different proteins that make up the receptor. And so you're going to get phosphorylation rather than that cross phosphorylation. You're going to get phosphorylation on one side, and that's going to activate the kinase, which will phosphorylate SMAD2 or 3, which is over here. And that's going to change the conformation. Here you have a conformational change that occurs. And that allows for interaction with SMAD4. And then it can go into the nucleus, and it can recruit other gene regulatory proteins and activate transcription. And so it's a latent gene regulatory protein. And what I mean by that, actually I'm not going to tell you. I'm not telling you anything else today. That's it. We're done. No. Okay. We're not done. We're never done. You'll have dreams about this. I hope not. What is a latent gene regulatory protein? So now I'm telling you, look, SMADs. Yay, SMADs. And I'm saying SMAD4, that's the brown one over here, is an example of a latent gene regulatory protein. And I think I put that on the quiz, maybe. So hopefully you've had some time to think about what that concept is. So what is it? How is SMAD4 an example of a latent gene regulatory protein? Yes?
Well, because it's already synthesized, and then when interaction after the signal, it gets to function as a transcription factor.
So it's already synthesized, and after signaling, it gets to function as a transcription factor. Okay, who wants to add to that? I want more. That's good, but I want more. So what makes it latent? Yes.
It depends on the degradation. It's inactive usually.
Interesting. So it depends on degradation of inhibitors. Are there any inhibitors being degraded here?
Okay, so what happens when it cleaves the cytoplasm?
Okay, so I love this because so what I'm hearing here is that you looked at one or two examples of latent gene regulatory proteins and you made it a rule in your head. Don't do that. I want you to be a little less specific. So what I got is something needs to be degraded or cleaved to activate. Yes, there are examples of latent gene regulatory proteins that are inhibited that way, and you have to remove their inhibition in order to activate. In this case, you have a latent gene regulatory protein that's also inhibited, but you remove the inhibition a different way. So a latent gene regulatory protein is something that's present, but it's present in an inactive state. You emailed me about this, right? Yes. It's present in an inactive state, and something needs to change to activate it, and sometimes it means it's degrading an inhibitor, sometimes it's cleaving something, and then in this case, it's just about a conformational change because they're in one conformation when they're inactive, and then once you get this phosphorylated SMAD2/3, then it can interact with SMAD4, and you get a change in conformation, which is kind of similar to JAK-STAT in a way. and then you get entry into the nucleus activation. Yes?
So, is SMAD 2 or 3 somehow, when it attaches to the SMAD 4, like exposing the NOS?
So, I like it. So you're thinking exactly how I want you to. So does SMAD 2 or 3, when it binds to SMAD 4, does it somehow expose an NOS? I do not know, but I would say you're probably right. Should I make that a stump the teacher question? Okay. So the question is, everyone's like, oh, I better wake up. She might give out a point. Okay. Oh wait, do I still have to put up points from the last one?
Yeah.
Someone email me and remind me. Okay. You guys have to stay on top of me. Okay. SMAD 3. Okay. So the question was, well, the hypothesis was, SMAD 2 or 3, when it binds to SMAD 4, does that conformational change somehow expose an NOS? Because how else is it getting into the nucleus? And so you need to, and so this is where I need to tell you my new rules, not new, but just I forgot to tell you them before. You cannot just copy paste something and send it to me. You need to actually summarize where your thinking is with a reference. Because I get a lot of like copy paste from wherever. Sometimes you might be right, but I'm not sure if you truly understand it. So someone somewhere probably identified an NOS somewhere. That must be, I mean, I think you've got to be right. How else would it get into the nucleus? But show me the data. So that means I need to see data. Yes?
Just referencing SMAD or any.
This is talking about SMAD 2, 3, 4 dimer. Oligomer. Okay? Okay. So that's my introduction to latent gene regulatory proteins. I actually was going to kind of, you got this question already before. So the question I was going to ask you guys was, what assumptions can you make about SMAD 2, 3, or 4 in terms of nuclear localization? But we don't know the answer. So you already got there before me. Okay. So signaling pathways that depend on regulated degradation or proteolysis to control activity of latent gene regulatory proteins. So we talked about latent gene regulatory proteins. They're just proteins that are involved in regulating gene expression, but they're latent. By latent, that means they're in some kind of inactive form, and they can be activated through multiple ways. And one way is through regulated degradation or proteolysis. So, and you guys already off the top of your head should have examples, right? Actually, I'm going to make that go away, and I want you to tell me off the top of your head, what pathway has a latent gene regulatory protein that's regulated, that activity is regulated by regulated degradation of something? NF-kappa B, there you go. Great. What's another one? Wnt. Okay, good. So I'm glad recitation is doing something for you guys. Good. Right. So the examples I'm giving you and that are straight out of the textbook, and you already went in NF-kappa B, were right on off the top of your head, which is great. And then the other one is Notch. So Notch is involved in development, cell fate, pattern formation, in development of tissues, and Wnt is involved in development, and NF-kappa B is involved in stress response, immune response, and all of them are somehow activated or regulated by either degradation of themselves, an inhibitor, something like that. So we're just going to go through that. We'll start with Notch because that's the new one. So this is involved in lateral inhibition in development. So this is where during development, one cell is going to turn into something. It's going to take some sort of developmental path, and it's going to tell the other cells around it not to. Lateral inhibition. So normally you'll have notch and delta, and so delta will be it's one membrane bound protein. It's an inhibitory signal that's expressed by the protein that's expressed by the cell that's developing into whatever pathway, and it's saying to all the other cells around it, don't do this. And it's going to interact with notch, which is the receptor protein on the neighboring cell. And so the way it works, this is involved with cleavage. So rather than degradation, you have cleavage of something. And so it's a latent gene regulatory protein because it's always there. It's just in an inactive form. And the way it's activated is, well, so you'll have your receptor. Well, remember, where do you guys think that the notch receptor is translated? Where in the cell? ER. You guys are on it. Yay. Yeah, so remember it goes ER, Golgi, plasma membrane. So actually, remember when we were talking about what happens in the ER, what happens in the Golgi, one of the things that happens in the Golgi is processing of proteins into mature forms. And so you'll get translation of this protein right into the ER membrane, and then it'll move by vesicular transport to the Golgi. And then in the Golgi lumen, you'll get cleavage right here at this number 1. And it'll just interact. So it'll be cleaved, so the protein, the actual polypeptide chain will be cleaved, but the proteins will still interact. And then it will move towards the cell surface. And so this will be the extracellular domain, right? And then when it interacts with delta, and that was on the cell that's saying, hey, don't follow me, don't do what I do, then it's going to interact with delta, and that's what's happening up here. And that's going to encourage cleavage. So that's going to, first it's going to get endocytosed into the other cell, and then you're going to get cleavage of this. It's going to expose this cleavage site, this number 2 here, and then that's going to activate cleavage here. And the whole point is really to free the tail, the cytoplasmic tail of notch, from the plasma membrane, and that's kind of your transcription factor in a way, because it's going to go into the nucleus, and it's going to bind to another protein that's bound to the DNA and activate gene expression. Okay, so normally there's a repressor here, and what happens is when the notch protein goes into the nucleus and binds to the repressor, it changes its function into an activator. So activates gene expression. And so in this case, it's a latent gene regulatory. How is it latent? How is it inactive? How? Come on, guys. I'm going to start calling random people and finding out who's not here. Ashley. Oh, look at that. You were raising your hand anyways.
Isn't it active when it's still attached to the notch on the extracellular side?
So is it inactive? So you're saying it's inactive when it's still attached to the, what's this? What's this membrane? Right, plasma membrane. That's how it's inactive, because it's stuck on the plasma membrane. So it's not about this other cell. It's not about anything having to do with the signaling cell. It's just the signaling cell, all that does is it removes this outside part, which exposes the second cleavage site, which allows then for the third cleavage site to be cleaved. So normally it's inactive because it's stuck in the membrane. And it's not until that's cleaved that it can be active. So I want you always to be thinking to yourself, wait, how is this activated? If this is a latent regulatory protein, it's activated through cleavage. Okay, any questions about this? know, guys. Someone said something about how there's only a certain number of weeks left of the semester, and it was a low number. Did you believe it? Something like 7 weeks or something.
It's like 6 weeks left.
It's like 6 weeks?
Last Thursday was the middle, exact middle
How
of
is
the
it possible? Have we known each other this long?
Feels like
Yeah.
in a good way.
So guess what, guys? Today is my birthday. And so I forgot about it. I'm like a horrible, no, don't do that. I'm one of those horrible, horrible birthday people who forgets everyone's birthday, including my own. And so this morning, now I remember it though, because this morning I was checking to see if the milk had gone bad by looking at the date. And I was like, oh. And so I went over to the calendar and I'm like, and then I realized that it was my birthday. Oh, it's really sad. That's what happens when you have kids. You forget about yourself. And you're worried about milk going bad. But anyways, I have a feeling there's going to be a cake in my future. My parents made a cake the other day, and there's going to be another cake because there was whispering that, well, don't go buy, because I always used to throw my own birthday parties because I forget everyone's birthday, like my poor parents. I forget everyone's birthday. And so I learned at one point, like, not to expect people to remember my birthday. So I'd always throw a big party, but now I forget my own birthday. So, but so my husband said, don't go getting a cake for yourself today. Good thing, because I would have forgotten. Anyways, so I'm older today than I was yesterday. I know, I'm always older. I'm always older. I thought we needed a moment to stop thinking about Notch. So I thought I'd share that intimate detail of my birth with you. Okay. What's next? We're going to talk more about latent gene regulatory proteins. No, seriously, milk. Milk was still fine, just so you know. Until the 24th. And that's just a cell line. Okay, so you guys, this is old news, the Wnt beta-catenin pathway. You guys know this one well because we did it in recitation. But this is another example of a latent gene regulatory protein, but kind of not really, right? So here it's like, it's inactive. It's always being made, but it's inactive because it's always being degraded, right? So that's how it's kept inactive, by degrading it constantly. And how is it activated? Something about GSK3? Yeah. Right, so GSK3 normally is phosphorylating beta-catenin, which allows it to be degraded. But it's when Wnt binds, then GSK3 is sequestered in MVVs, and that allows beta-catenin to be stable and build up high enough levels where it can get into the nucleus, activate gene expression. Apparently by kicking off Groucho. Who thinks that's funny? Groucho. And now one day I'm going to name my own gene. We were all born in a horrible time when a lot of genes have been named, right? If we had all been born like 20 years ago. Oh wait. 40 years ago. Okay, I'm all about birthdays today. Okay, so this is just showing. Okay, so before I worked here at Towson University, I worked at this company writing reviews for marketing purposes. And so I actually did this for their website. And so this is just showing the GSK3 sequestration in multivesicular enzymes. I always like to put that up there as a shout out to my peeps at SA Biosciences. By the way, well, I'm not going to say anything.
No, you start.
You know, if I went on the website for this company the other day, feel free to go check it out. You know, they were very good to me for all 4 months that I worked there. But, so they like killed me. I had to write a different review every week on a different signaling pathway, once per week. So imagine a term paper due every Friday, right? And it was horrible. It was horrible, but I did it. And so the person they hired to take the job after me has done like 4 in the last 3 years. And I'm like, how did she get away with like not like the back-breaking writing? Anyways, okay, sorry. Moving on. Okay, so NF-kappa B, another example of a latent gene regulatory protein. And how is it latent? How is it inhibited? IKB, right. How is it activated? Degradation, ubiquitination, and degradation of IKB, exactly. And that allows for liberation of NF-kappa B and the nucleus that goes. Right. Okay, so this is kind of a wrap-up, a quick wrap-up. No, it's not a true wrap-up. Don't worry. There's more. I just want to take a moment here to kind of go over a few things. So we did GPCRs, right? So they signal through G proteins and they regulate signaling through affecting levels of small intracellular mediators such as campin, 6-AMP and calcium. And then they do activate other things too, but that is unique to GPCRs, right? RTKs, they have the intrinsic kinase activity, and they activate other kinases through phosphorylation to relay the signal to the nucleus, right? And then there are the kinase-associated receptors, which are similar to RTKs, but associate with a kinase rather than having their own kinase domain. And then we just spoke about signaling through regulated proteolysis. So this is protein cleavage or degradation that liberates a transcription factor or inhibition of degradation, which stabilizes a transcription factor. And so I just kind of, I was trying to figure out how to categorize things. And so I thought these were nice categories of things we've spoken about. And so whenever you're thinking about the latent gene regulatory proteins, think about what keeps them inactive and what activates. Kind of try to organize it that way. Okay? Okay, so let's just like go back to basics for a minute. These are just some thoughts and concepts for you to be asking yourself while you're studying, because it's always easy to kind of start obsessing over details and not remembering the big picture. So just always ask yourself, okay, what's the purpose of this pathway or this whatever thing I'm studying? How is it a regulated process? All these things I've talked about with you are all things that are regulated, right? Because if it's not regulated, then what happens? I love it. Yeah, bad things happen, like cancer. And so you always want to ask yourself, why is it regulated? Like, what would happen if it's not? How is it regulated? Why is it regulated? And what would happen if it became unregulated? And how could it become unregulated? So you can look at every single part of a pathway and see how something can go wrong that could cause it to be unregulated. Like one example we used was RAS, right? If RAS is mutated in such a way that it's stuck in the on position, then that's really unregulated, right? Or PTEN was the phosphatase, right? The phosphatase that is important in dephosphorylating the PI3 kinase pathway, right? And so that's important in turning off signaling. And so if that's mutated and that doesn't work, then you're going to have dysregulated signaling. So pretty much every step of the way, there's a level of regulation. And it mostly has to do with phosphorylation or dephosphorylation or something binding to something or something being stable or degraded. So hopefully you're picking up on these things. Okay, so, and this should feel like review because it's so similar to what we spoke about with endocytosis. Because when I introduced endocytosis, we talked about receptor-mediated endocytosis as a way of turning off signaling, right? So signaling needs to turn off once the goal of the pathway is accomplished, right? So if there's an immune response because there's a virus around, you need to turn it off once that's taken care of. And so one mechanism, the major mechanism of turning off the pathway is inhibition at the receptor level, right? And so these were things we spoke about with receptor sequestration, receptor downregulation through endocytosis, inactivation through recruitment of some protein that was like with arrestin, right? Inactivation of a signaling protein, so you can, RGS hydrolyzes GTP to turn off GPCRs. Then there was also, what was the other one? Oh, this is in the practice problems about PKA, phosphorylating GPCRs to turn off signaling. That would be production of an inhibitory protein also, right? So sometimes you might have, so NF-kappa B, a big product of the NF-kappa B pathway, right? So remember NF-kappa B is activating gene expression. One of the major gene products are I-kappa B. That makes sense, right? You want to produce a lot of your inhibitors so you can shut off quickly. But this is all inhibition at the receptor level. Right, so there was receptor endocytosis as a mechanism for desensitization, beta-arrestin, binding receptors, incorporating into plasmin-coated kits, right? And of course, the minute you see arrestin, you should think of what other protein that has the same function? Adaptin. Adaptin, they both kind of are these ways to incorporate receptors into plasmin-coated kits. Receptor endocytosis, they can be recycled or sent to the lysosome, so that's receptor downregulation that flips to the lysosome. By recycling, you are pulling receptors off the cell surface for a period of time until they come back up. So that's also a form of downregulation. Not as severe as sending them to the lysosome. Sequestration in multivesicular bodies. You can also have receptor phosphorylation that inhibits signal transduction. And so that's kind of what we were talking about before with GPCRs, right? So here you have a hormone binding to a GPCR, and then that'll increase the amount of cyclic AMP. And then once you have very high cyclic AMP, that'll activate phosphorylation of the receptor, and that will inhibit signaling. And so in this case, the receptor is phosphorylated with a cyclic AMP-dependent protein kinase. So it's the cyclic AMP itself that's activating the protein kinase, and as cyclic AMP increases, the activity of that kinase increases, and you get shutting off of your signaling. We talked about RGS, hydrolyzing GTP to turn off GPCRs, right? Not only RGS, but also just binding to the target protein will also induce GTP hydrolysis. So this is all at the receptor level. I'm kind of flying through it because we've talked about it before. I don't think it deserves that much time. And so then I was trying to find an example of things that were not at the receptor level. And so I thought this was really nice. The JAK-STAT pathway can be turned off through inactivation of the receptor-associated kinase, JAK, through two different mechanisms. You can get degradation or inactivation of some of the, so actually Jack itself binds to ubiquitin ligase and can be ubiquitinated and degraded. And so if you get rid of this kinase, you're definitely not going to get signaling, right? Because that kinase is required for signaling. And apparently other components of the signaling pathway can be targeted for degradation. I think that might be it. Yeah. So that is it. I don't know if you guys have any questions. Wait, don't run away. We have like 10 minutes. We can talk about all sorts of things. Does anyone have any other questions before you run away? I flew through that, but I felt like we kind of covered it. Okay. Okay.